Hello, in this video we will discuss about the peroxisomal reactive oxygen species and metabolism. So the reactive oxygen species is very harmful for us in our tissue. But the peroxisome also present in the plant cell but not produce too much reactive oxygen species or produce, but it can be antioxidant. A lot of antioxidant present in the plant will inhibit. So here is the metabolism and the beta oxidation, embryogenesis, seedling growth, and in the animal the stomatal opening also, and in animal in liver hepatocyte beta oxidation will occur. So let's begin to understand the animal cell and plant cell. So this animal cell is here, you can understand, and in this way here is the plant cell, and the peroxisome is present in the yellowish color, while this is the cell wall and nucleus, and on the other hand the vacuole and the cytoplasm. This is the chloroplast used for the photosynthesis, but here is the mitochondria is the powerhouse of energy, also present in the animal cell, but the cell wall is not present, and chloroplast also, and vacuole is smaller as compared to the plant cell, and this is the plasma membrane only present in the animal cell, and peroxisome obviously present here. So this is the cytoplasm. So let's begin to understand the bigger picture of the peroxisome. So the bigger picture of the peroxisome I am drawing here to understand. This is the bigger picture of the peroxisome contain channels. So during the fasted state, the adipose tissue will convert in the animal to convert through a lipolysis from the adipose tissue. So this is the bilayer of phospholipid and the peroxisome. So the adipose tissue will produce the very long chain fatty acid through a lipolysis from the triacylglycerol to convert the glycerol as well as very long chain fatty acid, and this will convert with the acetyl-CoA addition through a CoA synthase enzyme and the very long chain fatty acyl-CoA formation. This is known as a VLCFA CoA. So it can be transported through ABCD1 transporter channel into the peroxisome. While the de novo lipogenesis, which that is convert the de novo lipogenesis convert from the glucose into the acyl-CoA, and this all will transport with a DMP70 transporter channel, branch chain fatty acyl-CoA, BCFA-CoA, and DMP70, which that is the bile acid synthesis intermediates also can transport through in this manner. So this is the VLCFA-CoA, which that will be enter, and the VLCFA-CoA, which that will be convert into the acyl-CoA, and the acyl-CoA with the acyl-CoA oxidase enzyme to convert the hydrogen peroxide, and enoyl CoA and 3-hydroxy-CoA will convert into the 3-keto-CoA, and it can be separated into the acetyl-CoA, and also the VLCFA-CoA again will recycle continue. So this acetyl-CoA will use for the Krebs cycle, and here is the D-amino acid oxidase enzyme will convert into the D-amino acid into the alpha keto acid and the hydrogen peroxide, and catalase enzyme can convert into the water and oxygen simply and easy. On the other hand, the reactive oxygen species will produce from the hydrogen peroxide. So in this way, if the hydrogen peroxide will not convert through catalase enzyme, hydrogen peroxide catalase enzyme. So here is the superoxide dismutase hypoxanthine also convert with the xanthine acyl-CoA. Xanthine oxidase will convert into the and reactive oxygen species are uric acid. So this is the synthesis the polyamine which that is ornithine, which that's come from the urea cycle, convert into the petrosine, and the spermidine and the spermine. The spermine, which that convert into the polyamine oxidase enzyme to convert the spermidine again, and also produce the hydrogen peroxide if the excess of the spermine will occur. So this is the hydrogen peroxide will catalase enzyme or peroxidase enzyme, which that convert into the water. But the reactive oxygen species also converge, and this reactive oxygen species will go outside from the peroxisome and damage the whole cell if the excessive reactive oxygen species will produce. Otherwise, it can be converted uh, through a through a catalase enzyme, and it is harmless. While here is the ATM. This ATM is the ataxia telangiectasia mutated, basically is the, you can understand with ATM. And here is the reactive oxygen species will trigger the ATM by the fixation of the PAX5. This PAX5 will fix and the phosphorylation of the ATM, and will give the donation of the phosphate group to ubiquitination of the trimeric ubiquitination. And in this way, here is the PAX5 will attach with the ubiquinone. So here is the PAX 10, 12 and 2 complex, trimeric complex will stimulate the phosphorylation. We also will lead to fixation of the P62 protein. And this P62 is the protein 62. And on the other hand, here is the reactive oxygen species with the dimeric complex TSC1, which that will be triggered by reactive oxygen species. And this reactive oxygen species will lead to RAB protein inhibition, and the AMPK will trigger this also like reactive oxygen species for the apoptosis. So this mTOR pathway which that inhibit because the RAB inhibit, and the uncoordinated 51 like kinase, the ALK1 which that will be triggered by the AMPK, and this ALK1 which that will be triggered by the reactive oxygen species, obviously due to the mTOR inhibition, and the paxophagy will be occur. This paxophagy means the peroxisome is involved for the autophagy. So this autophagy binding with the LC3 protein, and in this way here you can see the damage whole cell due to the autophagy, and the apoptosis will be occur. In this way, the peroxisome reactive oxygen species will produce, and the metabolism will be disturbed, and the whole cell will be damaged. And the whole cell will be damaged will lead to death of the cell is known as the apoptosis. And that is the significance and benefit of the peroxisome reactive oxygen species is apoptosis. Otherwise, the apoptosis will not occur, will lead to DNA damage and cause cancer. So thanks for watching. Bye.